my dear students, in this video we are going to understand about digestion and absorption of lipids. If you are new to my channel, please subscribe and share this with your friends. I also have the Telegram group, go and join it. The dietary consumption, first we have to understand which type of people consume how much amount of lipids as a part of their diet. Depending upon the economical status, the dietary habits will differ, right? If a person is from the underdeveloped country, he will consume less than 60 grams of lipids. If a person is belonging to the developed country, he will consume 60 to 150 grams of the lipids per day. The lipid composition. We have to understand that there are the different type of lipids which are taken into the body in the form of food. Most of the lipids include 90% of the triglycerol compounds. Along with that, phospholipids, cholesterols, cholesterol esters and free fatty acids. These all are taken into the body in the form of food. This is the overview of digestion and absorption of the lipids after consumption. We know that most of the lipids contain 90% of glycerols along with the phosphates, cholesterol and fatty acids. The adults consume 60 to 150 grams of lipids on an average per day. Depending upon economical status, the consumption quantity of lipids differs. First, we will understand the digestion in the stomach. The stomach have the enzyme called as gastric lipase. When a person is taking the food, it will mix with the lingual lipase which is present in the tongue. It is a type of the enzyme. It will interact with the minimum amount of digestion which will initiate the gastric lipase. This targets the fats and also the short chain fatty acids. These lingual lipase and gastric lipase will help the small intestine to digest the fats with the help of enzyme called as pancreatic lipase. In the adults, the action of the gastric lipase is lower, but in the intestine of the infants, these pH is neutral. At the neutral pH, the infant can able to break the milk fat. But in the adults, the pH is in acidic form, so the emulsification cannot easily occur in the adults, so that in the stomach there is less digestion or very minimum amount of digestion of the fats. First process is emulsification. When the food enters from the stomach to the small intestine, first process is emulsification. Emulsification is done with the help of bile salts, surfactants and peristalsis mixing movement mechanically. Here, what happens in the emulsification process? The process involves the dispersion of the larger lipids into the smaller lipid droplets. This will help in reduction of surface tension and also increase the surface area action. What is the outcome after the emulsification? Here, the end products are micro-sized emulsified lipids. That means large lipids are broken down into the stable emulsified droplets after the action of the bile salts. The bile salts are produced from the liver with the help of the cholesterol which is producing the bile salts. These bile salts and also the surfactants are acting in the emulsification process. This will accelerate the digestion process furtherly. What happens in the pancreatic digestion process? After the emulsification, the pancreas involved in the digestion. The pancreas will produce the enzyme pancreatic lipase and colipase. This colipase will help the pancreatic lipase to perform very well in the process of digesting the triglycerols which are also called as fats. What happens at the position of the 1 and 3 of the fats, that is triglycerols, they will cleave. That means the fats are broken at the 1 and 3 position. This will leads to the end product formation called as 2 monoacylglycerols and free fatty acids. I already said you the colipase is activated by the trypsin. These colipase will help the lipase for the further action of the pancreatic lipase. This colipase will inhibit the bile acid naturally for the proper functioning of the pancreatic lipase enzyme. What happens to the cholesterol esters? The cholesterol esters are broken down with the enzyme called as cholesterol esterase. These cholesterol esterase will break the cholesterol esters into the two end products called as cholesterols and free fatty acids. This is a enzyme specific digestion. What happens to the phospholipids? The phospholipids are combined with the enzyme called as phospholipase 2 enzyme. This enzyme will break the phospholipid at the position of the second position. Here it will lead to the final end products called as lysophospholipid along with the free fatty acids. These two compounds are formed after the breakdown of the phospholipids. This enzyme is inactivated by the zymogen, and this enzyme is activated with the help of the trypsin. This is the overview. Here we have to understand the fats are broken down with the enzyme pancreatic lipase, results in the formation of 2 monoacylglycerols along with the 2 free fatty acids. Here you can see the structure. At the position of the 1 and 3, these free fatty acids are broken down, leading to the 2 monoacylglycerols as the end products. Here you can see the picture. The food enters into the mouth. In the mouth, the food contains these type of the lipids. These all lipids are being unchanged in the mouth. They will enter into the stomach. Here also they are unchanged. After that they will enter into the small intestine. In the small intestine there are the enzymes. These enzymes will break these compounds into the simple compounds called as monoacylglycerol, free fatty acid, glycerols and cholesterols. These are the end products formed because of these enzymatic actions in the small intestine. This is the overview of the digestion of the lipids. Next we have to understand about some of the lipid esters which are left over in the small intestine, such as monoacylglycerols, cholesterol esters and vitamin esters. These are the compounds which are also found in the food. These are broken down with the help of the enzyme called as lipid esterase. This results in the formation of the free fatty acids. These are also requires the bile acids for the activity of the lipid esterase. After the process of digestion, there occurs the absorption. Here, the mixed micelles are helpful for the absorption. That means from the intestinal lumen, that is from the intestine, they will enter into the intestinal mucosal membranes for the absorption of the lipids into the bloodstream. So this process is called as absorption. It is acting with the normal diffusion process. That means these lipids will cross the mucosal cells and mucosal membranes, leading to the absorption. Along with the lipids, they will cross the plasma membranes, taking the vitamin A and vitamin K. These are the fat soluble vitamins. They will combine with the fats and they will enter into the cell membranes of the mucosa, which is present in the small intestine. Absorption efficiency. The bile salts are helpful for the absorption, right? The more amount of bile salts, the more amount of absorption occurs. We have to understand that monoacylglycerols and free fatty acids are the main compounds which are easily absorbed in the body. But only less than 40% of the cholesterol is getting absorbed into the body. And also there is an important point that short chain fatty acids and medium chain fatty acids don't want any action of bile salts. They do not require the micelle formation which is done by the bile salts. They directly get absorbed into the intestine itself. This is the overview of absorption. Okay, we have the bile salts, right? These bile salts will attach to the monoacyl glycerols and free fatty acids along with the phospholipids and cholesterols. These all compounds get attached to the micelles, leading to the formation of mixed micelles. These mixed micelles enters into the intestinal lumen. From here they will form the chylomicrons. From the chylomicrons they will enter into the bloodstream. From the bloodstream they will go to the liver, and in the liver there happens the metabolical actions. First we have to understand the digestion in depth. So here the digestion is done with the help of the digestive enzymes. We already know that the digestion is a process in which the lipids are emulsified and the surface area is increased, and the enzymes are getting acted on the lipids. They will break into the smaller compounds for the easy absorption into the blood. Initially, the enzymes called as the lingual lipase, which is present in the tongue, and gastric lipase, which is present in the stomach, they will help in the activation of the entire process of the digestion. Here you can see the picture. The food is taken into the body, the food enter into the stomach, the food containing the lipids. Here the food containing the lipids will interact with the bile salts. These will form the emulsified droplets. These emulsified droplets are acting with the enzyme called as lipase. This enzyme will break the large droplets into the smaller droplets. Here these are the small droplets which are monoglycerides, and these small droplets will combine with the bile salts. This will form the micelles. These micelles are called as mixed micelles. These micelles enters into the intestine. This process is called as absorption. These intestine mucosal cells get absorbed. Here again the micelles will disconnect. That means this micelle structure is destroyed. In the intestine the micelles will form the smaller triglycerides and some of the fatty acids. They again combine to the chylomicrons. The chylomicron structures are formed. These formed structures are entering into the bloodstream. From there they will go into the blood. From the blood they will reach the livers. This is the entire summary of the digestion. In the adults, the lipids digestion will not occur in the stomach because of the acidic pH. In the infants, the milk fat gets broken down in the gastric lipase because the pH of the stomach is neutral. This neutral pH helps the gastric lipase for the action of the milk. Okay, this milk fat is digested easily. The overview of emulsification. What is the emulsification? Large lipid droplets are broken down to the smaller lipid droplets. This will reduce the surface tension but increases the surface area. When the surface area is increased, the digestion is easily occurring with the help of the enzymes. So this is the big fat large globule is combined with the bile salts. This is emulsification. This leading to the formation of the emulsified droplets. You can see this, the hydrophobic side and hydrophilic side. This is water soluble, this is water insoluble. The three mechanisms of emulsification. For the process of emulsification, there are the three ways. First way is they undergo detergent action with the bile salts. Next, surfactant action by the degradation of lipids. And next, the mixing. Mechanically, this is mixed in the intestine. First, we have to understand the detergent action of the bile salts. Here, the bile salts and bile acids, they act interchangeably. Sometimes this will act and sometimes this will act, depending upon the pH which is present. The bile salts, how the bile salts are made? They are made up of the cholesterol. This cholesterol is produced in the liver. So these bile salts are having these type of the acids. Don't go in depth, only study the basic concepts. It will be asked in the entrance exams. Next one is the detergent action. Okay, in the detergent action, here what happens? The lipid molecules will combine with the some of the water contents, and this will lead to the formation of the fats into the smaller droplets. Here this is another detergent action where the emulsification of the fats occur, leading to the final small droplets formation. Next one is surfactant action. Here we have to understand some of the lipids like the phospholipids are emulsified by the surfactants. The surfactants have the hydrophilic end and hydrophobic end. What happens here? Some of the lipids, some of the phospholipids, some of the free fatty acids will attracted by the detergents. These detergents will attract the water molecule here. This water molecule, and here there is a oil molecule. That means here it is water soluble and here it is water insoluble. Likewise, these surfactants will attract the lipids to form the final micelles. This is another process of the emulsification. Next one is mechanical mixing. Our intestine walls will undergo the peristalsis movement. After peristalsis, mechanically mixing of the contents occur, leading to the fats. The big fat molecules broke down into the small fat molecule. This is another process of emulsification. After the emulsification, what happens? The pancreatic digestion. Our food contain the fats, cholesterol, esters and phospholipids. These all are present in the food. They are to be digested in the body. Here, first we will understand the action of colipase. Think, this is a fat globule. This is a small emulsified droplet. After the emulsification, the colipase will attach to the fats. Okay. These colipase will help the lipase to get attached. So this lipase is attached with the help of the colipase, leading to the complex further breakdown and digestion. So colipase is a type of the protein which will help to inactivate the bile salts and activate the pancreatic lipase. Okay. Next, what is the role of the pancreatic lipase? This lipase is an enzyme which digests the fats. The fats are broken down at the position called as 1 and 3, leading to the end products like 2-monoacyl glycerol and free fatty acids. They are the end products which are formed. So this is the picture. This is an enzyme acting upon the fats called as triglycerols, leading to the formation of 2-monoacyl glycerol and free fatty acids. Next, the degradation of fats. First one is the 90% of the food containing the fats, which is the triglycerol. So whenever we take the fat in the diet, that fat contain 90% of the compound called as triglycerols. These triglycerols are acting with the pancreatic lipase, this leading to the formation of the free fatty acids. So this process requires the bile acids for the activation of the enzyme. What happened to the cholesterol esters? I will show you the picture. The cholesterol ester is the type of the fat. It is a type of the lipid which is present in our diet. This is acted with the cholesterol ester. This enzyme will break down them into the two separate compounds called as cholesterol and free fatty acids. These two compounds are formed upon the action of the cholesterol esterase enzyme. What happened to the phospholipids? Phospholipids are acting with the enzyme called as the phospholipase. This enzyme will break at the position of the second of the phospholipids. These phospholipid molecules at the second position, they will break to release the free fatty acid and also release the lysophospholipid. These two compounds are formed upon the breakdown of the phospholipid. Remember the enzyme name which is breaking the phospholipids into the two compounds called as the fatty acids and lysophospholipids. After that, this phospholipase enzyme is inactivated by the zymosin, but it is activated with the trypsin which is present in the pancreatic juices. After the completion of the digestion, they should form the mixed micelles, right? I will show you the picture. So here, after the formation of the free fatty acids, here see, the lipids are combined with the bile salts. Here this process is called as emulsification. After the emulsification, there occurs the digestion. The digestion by the enzyme called as lipase. The fats are converted into the free fatty acids. The free fatty acids will combine with the bile salts. After the combination, this will form the mixed micelles. So this is called as mixed micelles. So these mixed micelles will carry the lipids into the lumen. So these mixed lipids will carry the lipids into the intestinal lumen, right? So the micelles are formed by the combination of bile salts and free fatty acids, right? These micelles will help as the vehicle for the entry of the fats into the intestinal lumen. After that, these triglycerides will combine with the protein compounds, leading to the formation of chylomicrons. From the intestine, they will enter into the bloodstream. This is chylomicron will help the transportation of the lipids into the bloodstream. From there, they will enter into the liver. So we have to understand the absorption. I hope you understand the digestion. Now we will study about the absorption. So absorption is a process where the lipids are entering from the intestinal lumen to the mucosal membrane. So this process is absorption. So the micelles enters into the lumen, right? So this process is the main absorption. The monoacyl glycerols and free fatty acids are highly efficient. That means they are easily getting absorbed into the body. But only less than 40% of the cholesterol is getting absorbed. Complete absorption, the monoacyl glycerols and fatty acids completely absorb. But incomplete absorption is the cholesterol. Cholesterol will only absorb up to the 40% only. But also the vitamins are getting absorbed into the body. Vitamin A and K are easily absorbed. This is the picture where you can understand the absorption. Here I said you, the bile salt will combine with the monoacyl glycerol, free fatty acid, phospholipid and cholesterol. These all are combining with the micelles. This mixed lipid is formed. After the formation of mixed lipids, they will enter into the intestinal mucosa. Okay, I hope you understand this. What is importance of the bile salts? The bile salts, this one is the bile salt, okay. The bile salt will get attracted with the triglycerols and also the lipase. This all combines with the bile salts for the formation of mixed lipids. I have shown you the mixed lipids, right? So these are the mixed lipids. These are the bile salts. Bile salts will combine with all the other simple fats to form the mixed lipids. That is the importance of the bile salts. What is the other importance in the absorption? These bile salts are helpful for the only absorption of the monoacyl glycerols and fatty acids. Only these compounds will get absorbed. But what happens to the short chain fatty acids and medium chain fatty acids? These short chain fatty acids and medium chain fatty acids does not require the bile salts because they directly easily absorbed into the body. They do not require to form the micelles. So this is the entire process. I hope you understand. Please share this with your friends and subscribe to our channel. Thank you, my dear students.